And again, this is from lab, the cell cycle. Your textbook goes into a little bit more maybe change of names. And so what I've done is if it's listed in the textbook slightly differently, I've put it in small parentheses. So for example, well, first of all, let's go back. You're responsible for the way that the lab book teaches it. I'm fine with that. You don't have to learn both terminologies on this one. 99% of the time, your cell is doing its thing. Your liver is detoxifying things. Maybe your liver is making some proteins. Maybe your liver is one specific protein that he's in charge of that's very important is your clotting factors. So if that's what your liver cell's major job is to do, then 99% of the time, that's what he's doing. But 1% of the time, we need that one cell to go through mitosis and make two more. Maybe that liver cell's getting tired. Who knows? You got to replace it. The liver also is amazingly regenerative. It is the most fascinating organ. So 1% of the time, we're going to go through mitosis, which is essentially the division of one cell into two daughter cells. This is when I told you that DNA is in the chromatin. Interphase is when DNA is in the chromatin state. It's thin, it's all over the nucleus. You really can't see it. If you look at a cell, maybe you could see it with electron microscopy where it's very, very tiny, but just our regular old microscopes that we use on a regular basis, you shouldn't see the DNA. It may be a haze of something going on in there, but it should not be bold and obvious. Okay, and again, what I tell people in my lab section is that it's like a kitten that has batted a ball of yarn till it's not a ball anymore. It's just thin strands of DNA everywhere. When it is time to go through mitosis, though, we need that chromatin to condense into a stick. So your strands of DNA become sticks. Remember, you have 46 chromosomes in every one of your cells except for your gametes, which are sperm and egg. 46 chromosomes, 23 from mom, 23 from dad. So every cell has 46. When it is time for that liver cell, I'm just going to stick with liver cell because it's easy. It's time for that liver cell to make daughters. You have to double everything before you can split that cell in two, right? You have to double the DNA. You have to double the organelles. So before mitosis happens, so therefore at the end of interphase, you have to replicate the DNA as well as the organelles, and we'll get to that soon. Okay, so I said we'll go through it in a minute. We'll go through it right now. DNA replication. Okay, so do you remember I said that the purines and the pyrimidines are linked together when we did the nucleic acids in chapter 2? They're linked together by a hydrogen bond, which is good because hydrogen bonds are easy to break, and sometimes we need to replicate DNA, or sometimes we need to read DNA to make a protein. So we're going to have an enzyme called Helicase unzip this little guy right here, unzip the DNA. We're going to separate the purines from pyrimidines. And then we're going to have another enzyme come along called DNA polymerase. He's going to read what got unzipped. And he's going to recreate a new DNA molecule. So he's going to come along here and say, okay, I see whatever, a C, so I'm going to lay down a G. I see a G, so I'm going to lay down a C. I see an A, so I'm going to lay down a T. I'm just making this up. I'm not reading this, but you can. Okay. So the first step, initiation, is helicase unzipping the DNA. Elongation is when the enzyme DNA polymerase comes along and starts, now we have it unzipped, he's going to recreate on both sides that got unzipped. He's going to lay down new nucleotides. So adenines with thymines, cytosine with guanine. He's going to hit a final little code and stop, and now we have 2 DNA molecules. They are identical. They both, one side of them, both came from the parent. So now we have 2 identical daughter DNAs. We're still at the end of interphase. Because each one of those DNA molecules has a piece of the parent, I can't go back and show you the slide, but you should pause now and go back and look 2 slides before. That DNA molecule, the original DNA molecule, unzipped, and now at the end you have 2 new DNA, daughter DNAs. One side of that DNA molecule is the parent. The other side is new from the DNA polymerase. Both daughters are half parent, and that's called semi-conservative replication. One side of the DNA is old, one side of the DNA is new. This helps prevent boo-boos because we don't want boo-boos when it comes to replication. That's bad. Oh, look, there's that picture right there. So don't worry about replication bubble. I mean, if I haven't said anything, I didn't say anything about replication fork or anything like that. But notice, here's the original right there. Here's new one number 1 right here. And here's new one number 2 right there. I mentioned this in lab or lecture, I can't remember, but those nitrogenous bases are classified as pyrimidines or purines. Purines are adenine and guanine. Pyrimidines are thymine, cytosine, and uracil. DNA has thymine. RNA doesn't have thymine, instead it has uracil.
In principle, copying DNA, a process called DNA replication, is very simple. The 2 complementary DNA strands separate, and because each nucleotide can only pair with its complement, A with T and C with G, each strand can be used as a template to build a new complementary strand, producing 2 DNA molecules. In the cell, DNA replication is a little more complicated, but the principle is the same. For clarity, we have untwisted the double helix. DNA replication begins at specific sites called origins of replication. Proteins attach here and separate the DNA strands, forming replication bubbles which grow in both directions. Enzymes called DNA polymerases move along the template DNA strands and catalyze the elongation of new strands. Eventually, all the replication bubbles merge, yielding 2 identical DNA molecules.
Okay, some cells don't go through mitosis. I mentioned this in my lab. Your skeletal muscle, when you go exercise and you become Popeye, it's not because your skeletal muscle cells are going through mitosis. It's because you're building up more proteins called actin and myosin inside every cell that you have. Neurons, they also don't go through mitosis. Cardiac muscle doesn't go through mitosis. If they get damaged, they can get repaired, so they'll get fixed with a type of scar tissue. That's not very good for your heart, but anyway, that's how you end up having heart failure from having heart attacks. But some cells need to continuously go through growth or go through a repair process, especially epithelium, as you will see in tissues, chapter 4. Okay, mitosis is going to be over with cytokinesis, because that's when you finish dividing at the end of telophase. You've completely divided the cytoplasm also. So cytokinesis, division of cytoplasm. Okay, don't worry about the term M phase. Let's just remember mitosis. This is when we're going to divide the cells. We're going to make 2 daughter cells. And remember, before you can go into mitosis, you have to double the organelles and double the amount of DNA. So you've already done that at the end of interphase. Now we're going to move on to the next 4 phases. Okay, so we doubled the DNA in the end of interphase, but for cell division, remember I said chromatin needs to condense into chromosomes. You've doubled them. So what's going to happen is your 2 pieces of DNA, so the 2 daughters, are linked together by a centromere. Those sticks look like an X because they're stuck together in the middle by a centromere. So the 2 sticks are sister chromatids. Together, it's called a chromosome. You remember the centrosomes? The guy that's going to put up those little spindle fibers? Those guys are an organelle. So those guys were doubled at the end of interphase 2. And they usually are going to be hanging out by the nucleus. But now, because we need to divide this cell into 2 daughter cells, one of the centrosomes is going to go to one pole and one of the centrosomes is going to go to the other pole. And they're going to start weaving their little strings of spindle fibers. Also, what you're going to see is the nuclear membrane, the nuclear envelope, is going to start to disappear because these centrosomes, these chromosomes are actually going to kind of spill throughout the cell. So as the DNA, the chromatin, starts to clump up, it actually starts to look kind of like a bunch of worms inside that cell. And they're not contained quite so beautifully in a little circle anymore. You can see that nuclear envelope's kind of gone. And there is a centrosome, there's a centrosome, and the little yellow things are the beginnings of spindle fibers. Now what's going to happen is all 46 of those chromosomes are going to line up in the middle. So this picture just has 4, but remember, people have 46. Maybe this is a salamander or something, but anyway, who knows? So metaphase, they line up in the middle. Metaphase, middle. Anaphase is where we're going to pull those sisters apart. So those spindle fibers are going to grab a hold of each of those sisters and pull them apart. So now what was one chromosome with 2 sisters, now the sisters aren't linked anymore. So now inside that cell during anaphase, you actually now have 92 chromosomes. But that's okay because you're going to finish splitting that cell up into 2. 92 divided by 2 is 46, and yay, we're all back to normal. Also, anaphase is the time where we have karyokinesis. So again, from lab, karyokinesis means the division of nuclear material. Your DNA is your nuclear material. It's in a nutshell. And so we're going to pull all of that apart during anaphase. Finally, telophase is when we are going to start creating that cleavage furrow. So actually it's going to be it's like actin is going to start to pinch the cytoplasm. So now we have cytokinesis, division of the cytoplasm. And the little dimples on the side where you can see 2 daughters are starting to form. That is the cleavage furrow, those little dimples. Those spindle fibers are going to start to disappear because they've already done their job. Chromosomes are actually going to start to uncoil and become chromatin. You're going to start to get that nuclear envelope again, and things are fixing to go back to normal, back to interphase. We actually have checkpoints during all of this, so we have stop and go signals, and here's another place where things can go wrong. But sometimes, if there is something going wrong, sometimes we can stop that process and we can repair it. Okay, protein synthesis. So you're loaded one segment of DNA, or I guess I should say a DNA molecule has thousands of genes. And so these genes code for different things. And I've already mentioned this: your hair color, your eye color, your height, your shape of your face, your earlobes, can you twist your tongue upside down or not? These are all genetic markers, okay? So what we're going to do in lab, the second part of lab, is we're going to make proteins. So DNA holds the genes to make different proteins. And you have a bazillion different proteins in your body. So you have a whole bunch of genes, okay? It gets read out 3 letters at a time. Those are called 3 bases in a row, or called a codon. And that's going to help determine what amino acid you need to make the protein. Remember, a protein is a whole bunch of amino acids put together. There is a codon wheel both in Lagniappe and in your your lab book, and that can help you determine what amino acid that you need. You do not need to memorize that. If you have the old lab book, instead of being a codon wheel, it's a chart. You don't have to memorize either one. Okay. So, you've heard of RNA. It's a single strand, not double stranded like DNA. But what's its purpose? Well, we have different RNAs, and they all have different jobs. One of them you've already met. Ribosomal RNA is made by the nucleolus, which is inside the nucleus, which is going to be the ribosome, where you're going to make the protein. It's the workbench for the protein. Okay. So all RNA is made in the nucleus, but it all gets to leave the nucleus and go out into the cytoplasm. I mentioned this already. What's the difference between your RNA and DNA? RNA is single-stranded. RNA has uracil instead of thymine. And then RNA has ribose instead of deoxyribose. Ribonucleic acid instead of deoxyribonucleic acid. So let's learn about messenger RNA and transfer RNA, mRNA and tRNA. Messenger RNA, he writes down the genetic information that's on the DNA. He writes down the matchup to the D's, C's, the C's, G's, A's, and T's. He writes that down. He writes down the code. He writes down the message so he can take that message to a ribosome to make a protein. When he writes stuff down, that's called transcription. So maybe you've heard of a scribe, somebody that writes down medical records. A scribe is writing. mRNA is writing what DNA has to say. And he is going to take that information and bring it to the cytoplasm. So, DNA is going to be unzipped, and let's just say on this left-hand side, here's the order of what has been unzipped. T's, A's, C's, and G's. So messenger RNA writes down the code by matching it up with what it pairs with. A's pairs with T's, but in RNA, RNA doesn't have a T. So for example here, you've got thymine, T T's pair with A's. So RNA puts together an A. Then, so we're essentially creating the message. We're creating mRNA. So an enzyme comes and sees the A right here. He says, oh, what goes with A? T. But I'm making an RNA molecule. So now I'm going to lay down the nucleotide with uracil. The next thing he reads, cytosine. So what pairs with cytosine? Guanine. So now I'm going to add to my RNA molecule a guanine. And then, there you go, guanine, make a cytosine. This is going to keep going. You're going to end up with a really long chain of RNA. And he's going to leave the nucleus with his message of what he read from DNA. I'm going to skip this. I've already said it a whole bunch of times. So transfer RNA is floating around in the cytoplasm carrying an amino acid. And transfer RNA kind of, let's see if I can get that to work there. No. Let's try this. Transfer RNA has a place for the anticodon right here. So let's say that here comes here's the ribosome right here, and I've got my ticker tape of mRNA right here. And on this ticker tape, maybe I have a U, an A, and a C. Now he would have gotten that information from that mRNA, got that information from the DNA, right? He wrote down what DNA had to say. So DNA would have been an A and a T and a G. Okay, and of course this is going to keep going. There's going to be a whole bunch more, but we're just going to do one codon right now. So what would match up with the U? That would be an A. What would match up with an A? That would be a U. What would match up with a C? That would be a G. Okay. So this right here is the anticodon. This is the codon on the messenger RNA. This is the anticodon on the tRNA. Let's look up on that codon wheel what UAC is. And I don't have the codon wheel. I have the chart, but that's okay. Whatever you have, we're going to look it up. So the mRNA codon triplet, UAC, is going to be UAC tyrosine. So this guy is going to know that he is the right guy because he has the anticodon that matches up with this right here. And what is he going to have? He's going to be carrying, transferring a tyrosine molecule. So he's going to drop that off right here on the ribosome. And then here's the next triplet, and that could be A, C, that's a C, G. And I'm going to have another tRNA molecule with an anticodon for ACG. This is very hard to do. 1, 2, 3. Dang it. So what would be the match for ACG? It would be U, G, C. Now you can use your codon wheel, and I show you this so you can understand how he knows he's the right guy. He has to match up. His anticodon has to translate what the message has to say. So if I look for ACG in my chart, ACG is threonine. So this guy right here is going to be carrying threonine. And he's going to come and drop off that amino acid right here, threonine. So now we have a tyrosine and a threonine. And this is just going to keep happening all through the message. Whatever the message is, whatever those little codons are, it's going to be translated by an anticodon, and whatever this message is going to be what amino acid gets dropped off. That is translation. We're translating messenger RNA. So, protein synthesis. First, you're going to transcribe. That DNA information is read and creating a messenger RNA. So messenger RNA is created. It is the message of what DNA has to say. And then mRNA is going to leave the nucleus through a nuclear pore, go set up house on a ribosome, and its codon is going to be decoded by a tRNA, translating it, and the tRNA is the guy that drops off the amino acid. Very similar to DNA being created. There's lots of terms. Initiation. So RNA polymerase is going to be the enzyme that does all of the transcription. He's going to unzip the DNA. He's going to be the guy that aids in the creation of the little mRNA molecule, and at the end of the RNA, there's actually a stop codon. You'll see that on your codon wheel, and you'll see it on your chart. That's how we know we can stop reading what DNA has to say. Excellent videos. Love them. My favorite. Now, sometimes you'll notice that, like in the textbook, sometimes there's going to be splicing of the mRNA and taking things out. And I'm not going to test you on that. But kind of like when you make a movie, sometimes you have to take out little blurbs to make it just right. That happens with mRNA too. Can you imagine if things aren't corrected, that something could go wrong? You don't make the gene correctly. There's 20 amino acids, but depending on how they are laid down determines what protein is being made. So any sort of error in transcription or translation can end up with a problem in the protein. Translation, same thing. Going to initiate it, elongate. We're going to make those amino acids stick together by a peptide bond. Then we're going to hit a stop codon, and it's done. You've made your protein. So in a nutshell, you start in the DNA. He has the genes of how to make that protein. What do you need? In lab, we're going to make a hemoglobin molecule. That's what we're going to do. Or maybe we've already done it at this point, probably. So by reading what DNA has to say, we're going to create mRNA. That's the message. That is transcription. Then those mRNAs are going to go set up house on a ribosome, and because of its triplets, its codons, the tRNA knows which amino acid to drop off because he has the anticodon. Okay, moving on. Cell aging theories. These are theories, and so we don't know what makes aging happen, and so I'm going to skip it. This is an example of what happens when you get a defective protein. You probably have heard of progeria. It makes people look old when they're just kids. And so this protein is very important for normal growth, for healthy body, healthy hair. And if you have something go wrong with protein synthesis, that you don't have a normal progerin protein, you end up with this disease. That's how it gets its name, progeria. It's evident in a small child, and these people are usually deceased by age 20. And that's just a memorization thing. So here's a video.